Well, hello there. I get ready to record these and then I just sit in the chair and stare at myself and think, how am I going to do this? So I've been doing that for a little while this morning, and then finally just hit the record button and let's get going. So here I am with my tea. And well, I don't know. Do I want to give you guys a pep talk? Not necessarily a pep talk, but a statement that we're halfway through. This is a halfway point. Content's going to get a little harder. We're about to start the hardest chapter in the book. So once we're through it, it's only going to get a little easier, or I don't want to say easier, total downslope, but maybe a slight downslope, because this has anatomy that goes microscopic and has three separate cellular mechanisms that you'll have to learn. So it's a tough one. And usually I go really slow with this chapter. It's chapter 10. This is lecture 1 of chapter 10. And I usually take 2 steps forward, 1 step back, 2 steps forward, 1 step back, and spend a good almost 2 weeks in lecture on this material. But it's kind of weird to do that when I'm just looking and talking to myself. So my recommendation to you is to watch these videos more than once. There might just be 4, I don't know, we'll see. And we're just gonna dive right into it. So buckle up, all right? Yep, it's just weird laughing and talking to myself. Very strange. But I am thinking of you guys. I am wishing you luck on this chapter. Like I said, it's tricky. I have a beautiful sunny Sunday. I record these typically on a Sunday. And, when I'm done, I get the rest of the day. So there's a little incentive to me to just move ahead. So anyway, by the way, winter is coming. There's a G there. Really, it is coming. It's getting colder. That's my son's alarm clock. It's just lovely. Anyway, sorry, guys. You signed up for this. All right, well, let's get to the PowerPoint. And, of course, I have to itch my face because that's what happens. So we are talking about muscular tissue. And like I said, there's a lot. There's a lot in this chapter. Take it slow, pause, take a break, do what you need to do to chew on this material. Okay, let's see what we got here. So muscular tissue. Well, we're going to start, and you should be pretty comfortable with the initial information. When we did tissue types, of course I'm angled, now I can't reach anything. We had skeletal muscle, cardiac muscle, and here I am, smooth muscle. So when we were talking about these 3 different types of muscular tissue in the tissue chapter, I don't know if you remember, but we kind of made a table. And we thought, well, this is the difference between skeletal muscle, cardiac muscle, and smooth muscle. So skeletal muscle's location is across bones. All right, so location across bones. I don't know, skeletal seems weird. Cardiac muscle is found in the heart, and smooth muscle, I'm never going to call it visceral, is lining hollow organs. Such as the gastrointestinal tract, like the small intestines, but also blood vessels. Let's not forget that, because this is critical in terms of managing blood pressure. So skeletal muscle's function is to move bones. It is striated. It is multinucleate. Its shape is long, unbranching. Oh, so it begins. That's an O, by the way. Long and unbranching. Fibers. I'm going to use this word fibers, okay? A muscle fiber is a muscle cell, okay? That's what a muscle fiber is. And it's voluntary, meaning we can control it. There are times we don't control skeletal muscle in terms of reflexes, but overall we control skeletal muscle. Cardiac muscle is our long and branched, found in the heart. Function is to pump blood. It has a single nucleus and is striated and has these intercalated discs. Those are these little junctions. They're a type of gap junction because we want cardiac muscle to be coordinated. We don't want each cell to do its own thing. We want them all to be coordinated, and it's involuntary because we don't control it. That could always be up for discussion. I do agree, because if you can meditate, can you slow your heart rate? There's biofeedback mechanisms where people do try to manage their heart rate, but for the most part, it's your brainstem and your hypothalamus managing your heart rate. And then the last one is smooth muscle, lining hollow organs. Function to move stuff. One nucleus, so it's single nucleate, no striations. These are spindle-shaped cells. The more I say that, the more I understand that word. So fibers, I could say. And it's involuntary because we don't go, okay, English muffin, get out of my duodenum and head on to my jejunum. We don't do that. We don't have enough time. All right, what's the function? Why do I need muscle tissue? I need muscle tissue for body movements. Hey, nope, not onto special properties. Here we go. My pen wasn't working. I don't know why it's always red. I wouldn't mind it being a different color, but I don't know how to change that. And I'm just finishing up the semester, and we'll just keep it red. All right, so right, we move and we stabilize body positions. We don't even know that our muscles are always contracted to maintain a body position. Otherwise, we would be on the floor in a bony puddle. Move substances within the body, so movement outside the body, inside the body, stabilizing body positions, and then it generates heat. Because when you're cold, you shiver. I have a scarf on. It's actually pretty toasty in the house, but I didn't want to look stupid in a t-shirt, so I thought I'd dress it up a bit and throw on a scarf. And even a little makeup for a Sunday. You should be impressed. Anyway. And a byproduct of muscle contractions is an unusable form of energy, and that is heat. So our body says, well, hey, if we can't do anything with it, but we can take advantage of it if we're cold and actually make, where's my pen? We can actually make the body shiver, right, to help maintain my temperature, homeostasis. So sure, but if you're running around in the hot summer and you're still generating a lot of heat because it's a byproduct of muscle contractions, nothing you can do, then your body's like, oh, you're killing me now. I gotta cool down and have a separate mechanism to offset the heat generated by muscle contraction. So it's a good thing, and it's not a good thing, right? There's pros and cons. Right, so those are the functions. Guys, don't mess up functions with properties. This is an easy question on the test. What are functions of muscle tissue? What are some special properties? So we're going to skip the first one because that's actually the hardest. Skeletal muscle contracts, which means it gets shorter. It extends, which means that it gets longer without being damaged. Hey. It's just if I pen tip, I wonder. All right, let's see if I can. Oh. This is going to be a long lecture. Longer, it extends, and then it can return to its original length. So that means it has elasticity. It can get shorter, it can get longer, and it can go back to normal. So those are 3. And then the other one is this electrical excitability. This means that muscle tissue responds to movement of ions. It can and it can generate its own movement of ions. That's my son's alarm clock. That's what that is. That's totally making me lose my train of thought. But I'm not starting over. I don't know why I'm whispering. All right, Oh, so electrical excitability. Bottom line with that is there is a flow of ions, and there's a very interesting flow of ions associated with muscle tissue. So flow of ions is really electrical activity. So what's going on with muscle tissue is that one, it generates its own electrical activity. That's its impulses. And two, it's able to be excited by neurons, by somebody else's electrical activity. Can be excited. So stimulated, I guess that's what the slide says. Oh my gosh. By neurons. Right? That's kind of a big deal. Not everybody can. And we're going to talk about how it's actually a neuron talks to a muscle cell. It's one of the 3 special mechanisms in the neuromuscular junction, but that's not this lecture. This lecture has enough content. All right, so we're going to start big and look at just all this words. Look at this anatomy. So, I need a drink for that. All right, so the first thing we do is we take the actual skeletal muscle, like this guy's, whatever, taking this muscle in the lower arm. Right? Taking a skeletal muscle, we're only going to be talking about skeletal muscle other than what we originally talked about, compare and contrast the 3 types of muscle tissue. All right, so we're going to go in depth, and we're going to go pretty deep. It's a love story. It's my love story, actually. Well, one of my love stories. There's been more than one. But we'll get to that. Not in this lecture. All right. So we start with skeletal muscle. Skeletal muscle has its own covering, and its covering is called the epimysium. So I have a skeletal muscle contained with its own covering. Now, if I dive into this muscle, I find these bundles. A bundle of muscle cells, a bundle of muscle fibers is called a fascicle, all right? So a fascicle is a bundle. And this bundle has its own covering called the perimysium. So perimysium is around a fascicle. Epimysium is around the muscle itself. I'm going to try to write that word muscle. And I'm going to try one more time. So it's around muscle. All right, so the epimysium is around the muscle. Inside the muscle, there's bundles called fascicles. These fascicles have their own covering called perimysium. As I dive deeper, I come to a muscle cell. Muscle fiber, I told you I was going to call it that. And this muscle cell has its own covering called an endomysium. So endomysium is around a muscle cell. All right. So let's take a second. Let me get big. Let me get my little demo. So you're like, there's a lot of vocabulary, unfortunately. So here I have a plastic bag, okay? And in this plastic bag, what's in the bag? Nothing that interesting, sorry guys, are 2 boxes of spaghetti, okay? So let's take this. So this plastic bag is like the covering around my muscle. So this plastic bag is representing the epimysium around the whole muscle. Now if I dive into the muscle, I come up with bundles, right? So this is a bundle. That's upside down. And so this would be a fascicle. A bundle of muscle cells inside a muscle, and the box itself is representing the perimysium. So if I dive into it, I will pull out a muscle cell, aka a piece of spaghetti, because they're long, thin, and unbranching. So this is a muscle fiber, and this muscle fiber is surrounded by endomysium, all right? So those are the three coverings associated with a muscle. Epimysium around the muscle, paramysium around a fascicle, and endomysium around a muscle cell. All right, let me get little again. All right. So let's dive back to a couple of slides and look at some of the words. Okay, so I think we already covered this. Everything has nerves and blood vessels. That makes sense. I have fascia, which is connective tissue, kind of separating and uniting different muscles. We're not going to see fascia in this course. And then, if you remember, I have tendons that connect muscles to bones. So what's interesting are these three coverings, the epimysium, paramysium, and endomysium. I didn't know this until I actually read the book the other day. Well, I read it more than once. They all unite together, going past the muscle fiber and turn into the tendons to attach to bone. Right? So a tendon, it actually consists of, I always thought it was just epimysium, but it's all three. It's epimysium, paramysium, and endomysium. He will eventually turn that off. I swear. All right, so we had the epimysium, the paramysium, and the endomysium. Okay, so now we're ready to talk about a muscle cell, a muscle fiber. Oh, and look, here I talk about tendons again. Most muscles insert into a bone via a tendon, but there are a few that are an aponeurosis. We're not going to talk about those at all. We have nerves that go to muscles. I have blood vessels because muscles need a lot of nutrients and oxygen. Let's talk about this. Plentiful microscopic blood capillaries. It's always about capillaries because capillaries are the only blood vessel that can dump things off and pick things up. So why do I need oxygen? Needs constant oxygen and nutrients. Why? Because I have to make ATP. Okay, so if I have oxygen and nutrients, I'm going to make ATP very efficiently. If I don't have oxygen, I'm going to make it very inefficiently. Right? So it's very important to have good oxygenated blood going to my muscles. And we're going to talk about that later. That's probably going to be lecture 4, and we're only in lecture 1. Oh boy. All right. I don't care about development, but after birth, you can make skeletal muscles very large by adding more protein. Can't store excess protein, but if you can build muscles, you will use it. Or you can make muscles very small and atrophy because they lose the protein because of lack of use, for whatever reason. Oh, right. What do I want to talk about here? Okay, let me get to the muscle cell. So, this is another drink. So we're going to start with this picture that's right here. All right, guys? What we have is, what you're not seeing is the endomysium. The endomysium is around, I really want to just scream here, but I'm trying not to. Okay, so that is around a muscle fiber. I'm going to try to just use the word fiber for cell. All right. And this muscle fiber has very unique anatomy. So it has a lot of special names. So now we have to talk about what is in a muscle fiber, right? It's a cell. So it's got the cell contents of like a mitochondria. And what we're not seeing are the many nuclei But remember, skeletal muscle is multinucleate. So I have mitochondria, I have nuclei, but I have a very special plasma membrane called a sarcolemma. All right? So I have this sarcolemma, and then I actually have plasma membrane projections that run inside the cell. And this are called transverse tubules. So transverse tubules are projections of the sarcolemma. Inside cell, okay? And they're actually also known as T-tubules. And they're important. They're like hallways, all right? You kind of have like your outer walls of your house, but then you have hallways and it's all connected. So I consider T-tubules hallways. And then I also have very special storage areas. That's this tan stuff, and that's called a sarcoplasmic reticulum. So I call these closets, right? And these closets contain calcium ions. And that's actually very important. So contain calcium. I'm going to just say ions. I know you're not going to be able to read this, but so this is called the sarcoplasmic reticulum. Is like a closet. Okay. So I had my sarcolemma, plasma membrane. I have my T-tubules, transverse tubules, projections of the sarcolemma. I have my sarcoplasmic reticulum containing calcium ions, my closets. Oh, here's a nucleus. My nucleus, mitochondria, a lot of mitochondria because this is where ATP is made. You know what I'm trying to say. Now, inside, I hate talking about this one. Inside the cell are these long red guys, right? And they are called, they're protein strands. And they're called myofibrils, right? It's kind of a middleman. And myofibril protein strands have a very special banding pattern. This is the root of those striations we see in this cell. And these myofibrils, look, it's not on here, contain myofilaments. Ew, right? So myofilaments are contained within myofibrils. Myofibrils are in a muscle fiber. Muscle fibers make a fascicle. Fascicles make a muscle. Oh, I know I have that as a table. One last little vocabulary about the anatomy is sarcoplasm. Sarcoplasm is the cytoplasm in a muscle cell. Oh, I said I was going to call it a fiber. In a muscle fiber. Great. Let's see. So let's go back a couple. Go back to where we're comfortable. Muscle fiber development. I don't care. So I have the sarcolemma. That's the plasma membrane of a muscle fiber. I have the transverse tubules that are extensions, projections of the sarcolemma. This was like the hallways. I have sarcoplasm, which is the cytoplasm, and I have a whole lot of glucose, and we'll talk about myoglobin another time. I also have inside a muscle fiber myofibrils. This is what gives it the striations, the stripes. These myofibrils contain myofilaments. And then I have the sarcoplasmic reticulum. That's like a closet. I have like a closet door, terminal cistern. I'll never use that. Connected to the hallways. Okay, you're starting to see an image. And they are storing calcium ions. That is super important. Okay, it's part part of the love story. All right, so let's move on. Ooh, I guess we're there. Okay. This one's going to be tricky. Oh, we're doing okay with time. Okay, so let's orient ourselves. Where are we? We are talking about a muscle fiber, all right? So this is a muscle cell. Remember, we had the sarcolemma, plasma membrane, sarcoplasmic reticulum, the closets, the T-tubules, hallways. I don't really care about that other word. But then I have this myofibril. Oh, and of course, sarcoplasm. So this myofibril, these long protein strands inside the cell. You know, I usually don't even talk about myofibrils, but I feel like I need to because I'm not with you guys. Consist of myofilaments. And there's two types of myofilaments. Oh yeah, we're going there. There's a thick filament, because it's thicker. And there's a thin filament, because it's thinner. This is supposed to be the word filament. Okay, and they make up this pattern. So really, myofilaments are what make up this pattern of bands. This banding pattern that we see, and we say that skeletal muscle is striated. And well, that's what we're going to talk about. So this banding pattern is, we'll start down here, right? See how it starts here, goes from one place to one place, and then it just repeats itself all the way down. So this repeating banding pattern is called a sarcomere. All right, so this is the repeating, it's actually called the repeating unit of contractility. Remember we had the repeating unit of compact bone? This is the repeating unit of contractility. We're going to talk about what does it even mean to contract? How do they contract? What does it mean? Is a sarcomere. So there are two ends to a sarcomere, and both of the ends, it kind of defines the boundaries, are Z-disc. So Z-disc to Z-disc is a sarcomere. Right? So when we look in here, and then you see this other like blue straight line, that's the M line. So the M line right in the middle. That's how I look at it. M line in the middle. And I see that I have a thin filament, the lighter red, and a thicker filament, the darker red. So they've taken that and they've actually given it a slightly different look. So the thick filament here in the middle that is red, and then the thin filament, they've actually made this yellow here. So the thin, so that guy there is a thin filament, which I know it's already actually labeled, right? And this guy, all right, so that's a thin filament. And then the thick filament is blobby red, to blobby red, and that's the thick filament, right? So on this picture up here, thick filament is this thick red guy. It crosses the M line, the middle line. And the thin filament, kind of, it doesn't. It's on the ends. Thin filament is connected to the Z discs. Oh, let's write that down. Thin filament connected to Z disc. Thick filament crosses M line. I'm going to run out of room. It's all good. All right. Now, the length of the thick filament. The length of the thick filament is the A band. Right? So the A band is the length of the thick filament. Doesn't change. Will always be the same. The I band is where it is thin filament only, no overlap. So thin filament only, no overlap. All right. All right. So we've done the A band, we've done the I band, we've done the thick, we've done the thin. The other part that we have is we have the part of the thick filament where there's no overlap, and that's the H zone. All right. We're gonna write that so right over here. H zone is thick filament only, no overlap. Right, so I have a thick and I have a thin, and they overlap a little. The part of the thin where there's no overlap, so like from my wrist to my elbow, is I band. The part of the thick, so from this wrist to this elbow, if this is a thick guy, where it's thick only, is H zone. All right. That's a lot. But we're not done. Hang on a second. So let's go back here. Oh, this gives you all the words of what we just talked about. The functional unit of contraction is a sarcomere, and it's repeating. So the repeating functional unit of contraction. And it consists of myofilaments that are thick and thin. Z disc to Z disc kind of denotes a sarcomere. And I have the A band. This is a length of the thick filament. I have I band, where it's only thin filament, no overlap. I have the H zone, where it's only thick filament, no overlap. And then I have, the M line, which is in the middle of the sarcomere, and it is on, kind of holds, let's just say holds thick filament in place. All right, I'm running out of room. Okay. Oh. Did I talk about this? Oh. Yeah, I guess. All right, give me a second here because I want to talk about this, that, and then we got a little bit more to talk about, and then we're done. Okay. Let's go to that beautiful picture. I love this picture. It's silly, but it makes sense. This is a muscle fiber. All right, so I have the sarcolemma, the plasma membrane. I have the sarcoplasmic reticulum with calcium. I have my little hallways called my T-tubules. And then I have like the closet doors. I have many nuclei because I'm multinucleate. So this one has 3. I have mitochondria. Mitochondria is where ATP is made. I have this myoglobin. Myoglobin holds on to oxygen, kind of an oxygen storage. I have glycogen granules, so that is where I have some stored nutrients, because I need nutrients and I need oxygen constantly to make ATP in the mitochondria. All right, I have my myofibrils. I'm probably never going to say that word again. But inside of it, I have my myofilaments. I have my sarcomere. So this here is showing me 1, 2, 3, 4 sarcomeres. Are you right? And they're kind of held in place. They're held in place by many different other proteins. And one of the proteins is a dystrophin protein. So maybe you're like, whoa, muscular dystrophy must have something to do with that protein, and it 100% does. So do your paper on muscular dystrophy if you wanted to learn about that. It's a tough disease. All right. So in my sarcomere, the ends of the sarcomere are defined by the Z-disc proteins, and my sarcomere consists of thin filament and thick filament. In a very special, I'm repeating myself, yeah, I am. It's in a very special pattern, all right? So if this is Z-disc to Z-disc, that is one sarcomere, all right? I have the thick filament. Oh, it's labeled now. And the thick filament is myosin. I have a thin filament, which is actin, and they overlap, okay? They overlap, and it's very important that they overlap. But the area where there's no overlap, I guess here is where it showed. The area on the thin filament where there's no overlap is the I band. The area on the thick filament where there's no overlap is the H zone. In the middle, stabilizing the thick filament is the M line. And then I have the length of the thick filament, which is the A band. All right, but they overlap a little. A band will never change because it's the length of the thick. Well, you know what? And therefore it will never change. See, I'm going to go back. Length of thick filament. Oops. Haha, I see that. All right. It will never change. Okay, so my thick filament is my actin. My thick filament is my myosin. You can tell I'm tired. My thick filament is my myosin. My thin filament is my actin, and they overlap a little. So we're going to look at these myosin and actin a little bit more closely. Oh, look, this is what it actually looks like. How crazy is that? It's beautiful. Z-disc to Z-disc. Look at the colors, well, the black and white pattern of overlap. Okay, so we have some other structural proteins associated with a muscle fiber. We're not going into that, so you don't to really worry about these, but we have the two main players, which are actin and myosin. And then we have these other guys we're going to talk about in a minute, which are troponin and tropomyosin. Okay. So here's the thick filament. Thick filament is myosin, and on myosin, it has all these little heads, okay? And these are important, so those are all myosin heads. And it's on both ends. And then at the other end, it'll go all knobby. And then in the middle, you would find the M line, all right? Trying to build a story. So I have actin, and actin has these special sites that are binding sites for binding with myosin. And the second those sites are uncovered, myosin will bind with actin. It will bind. But those binding sites are covered up by this regulatory protein called tropomyosin. It's a strandy protein, and it's held in place by this other protein called troponin. So tropomyosin covers the binding sites on actin, so therefore it prevents myosin from binding to actin because it blocks it. Can't bind if the sites are covered. And troponin just holds the tropomyosin in place. Okay. I love these guys. They're so fun. This is part of a story, and we're getting there. All right, so this is just a little bit more about the myosin. So the myosin is the thick guy with these heads and tails. And myosin is what will kind of work with ATP. So ATP is actually talking to myosin. We'll just leave it at that. Actin is the thin one, and it has the binding sites for myosin, but they are covered by tropomyosin. And troponin holds the tropomyosin in place. I'm going to skip this calcium part here. Then we have structural ones, and we're not going to worry about that so much, but they are interesting. Chitin anchors the Z disc to the M line, holds everything together, stabilizes the sarcomere. Yeah. And dystrophin will link the sarcomere to the plasma membrane. All right, so just a quick little back up. We have muscles, skeletal muscles, attached to bones via tendons. They have the outer covering called the epimysium. Inside a skeletal muscle, there are bundles. And the bundles are called fascicles. And the fascicles have their own covering called a perimysium. Inside a fascicle, we will have the muscle fiber. So that is the actual muscle cell. All right. So when I talk about a muscle fiber, it has its own outer covering, which isn't shown here, called endomysium. Okay. So the endomysium is fused with the sarcolemma, the plasma membrane. I have my T tubules. I have my mitochondria, my nucleus, my sarcoplasm, the liquid part of the cell. And then I have my closets, my storage, my sarcoplasmic reticulum. That is full of calcium. Ions. Everything's an ion. Inside a muscle fiber, I have myofibrils. And my myofibrils are made of myofilaments. I have the thick myosin one. I have the thin actin one. And they overlap, right? This overlap is very important. All right, and we are going to stop there. See you in a few.